So I'd like to thank and congratulate Sunny for the kind invitation to this amazing conference. I've learned a lot. So today I'd like to share with you my personal journey in this field of circulating DNA. I studied my secondary school in Hong Kong, in St. Joseph College. And in there, we actually use a particular textbook called Biology, a Functional Approach. And this book is interesting because inside there's actually many photographs of famous scientists. And one of those is this pictures, which have been shown multiple times in this conference, which is a picture of Watson and Crick standing in front of King's College Chapel in Cambridge. So I remember when I saw this, I was thinking about Cambridge and how it actually trained generations and generations of outstanding scientists. So eventually I decided to actually apply to Cambridge. And fortunately, I actually joined Emmanuel College in Cambridge. And then I studied medicine there. But in Cambridge and Oxford in those days, for medicine, they only accept you for 3 years. And after that, you have to reapply. So then I thought, well, it might be a good idea to try the other place. So I went to Christ Church in Oxford. And so this is a picture of myself as a medical student in Oxford. So we have to study different subjects. And I remember, actually, one of those subjects is obstetrics. So I learned about how when a woman is pregnant, she would like to know the health, including the genetic health of the baby. But at that time, the most direct method is to stick a needle into your uterus and to take some fluid. But every time you do that, there's a risk that you might harm or even kill the baby. So as a young medical student, I was thinking, is it possible that we could develop a method which does not have that risk? For example, by taking a blood samples from the mother. So at that time, as a medical student, I did a couple of experiments, but then I qualified and as a houseman. But during that 1 year as a junior doctor, I was constantly thinking about this field. And so after 1 year, I decided to actually go back to Oxford and do this full-time as a PhD. And then we try different methods. So the question is, how do you actually do this non-invasive test? So I was thinking, maybe the baby would release its cells in the bloodstream of mother. But then how do we know that the cells is from a baby? Then I thought, if the baby is a boy, then maybe we can actually see male cells circulating in the blood. So we try a variety of methods, for example, by using in situ hybridization, and we're able to actually see a Y-chromosome signal, the black dots there, in late pregnancy. And then also at that time, the method of PCR has just been coming in. So actually in Oxford, we have a professor called Professor John Bell, who is actually one of the first person outside the CEDAS team to actually successfully implement PCR. So I asked him to teach me to do PCR, and then we had some success in detecting these fetal cells in mother's blood. But however, the problem is that the accuracy is just not high because the number of fetal cells in mother's blood is really low. It's like hunting a needle in a haystack. So after 3.5 years, my scholarship ran out, and so I have to really go back into clinical practice. So at that time, the science didn't go well, but at least actually I met my wife, Alice, and so this is our graduation photo in which we got our PhD in the same session. And then I worked a few years as a doctor, and then 1997 came, so that was a year when Hong Kong went back to the sovereignty of China, so my wife and I decided to actually go home. So I joined the Chinese University of Hong Kong. So you can see the campus here is in the northeastern part of Hong Kong. Is pretty beautiful. And I remember actually about 3 months before I actually left the UK for Hong Kong, in this particular issue of Nature Medicine, there were 2 papers which talk about in patients with cancer, you can actually find cancer DNA in the plasma and serum of the cancer patient. And then suddenly I have a strange thought. I thought the baby actually growing inside mother is a little bit like a cancer growing in a patient, except that in my clinical practice, I've never seen a cancer which is as big as an 8-pound fetus. So I thought that if my hunch is correct, I should be able to actually also see fetal DNA in the mother's plasma. So in other words, what it means is that in the previous time when I was a PhD student, I've been actually looking in the wrong place. I was looking at the cells, and what I should be looking at is in the plasma. But then the problem is that at that time, I just resigned from Oxford and don't really have a grant in a Chinese university yet. And so I really don't have a lot of resources. So I have to do something really very cheap and easy. But the problem is I've never actually handled plasma up to that point. And I thought, what can be very cheap and easy? Then I recall a day when I was in Christchurch in Oxford. I actually never quite liked the college food. So because of my so many years in Hong Kong, I like Asian food. So sometimes I actually go home and cook some Asian noodles. And of course, the way you cook the noodles is you boil some water, and then you put the noodles in. And a few minutes later, you have your meal. I was thinking, well, can I just take a few drops of plasma and serum and just boil it, and then take a few drops for PCR? And you probably think it's crazy. But interestingly, even with this very crude method, I was able to actually get some signal. For example, a positive signal there, a negative. And then later on, I find that all the women to give the positive Y chromosome signal all have baby boys, and all the ones without signal all have baby girls. So it's the first discovery of cell-free fetal DNA in maternal plasma and serum. And then we find that this DNA is present very early, like from 7 weeks of pregnancy. And actually, by the 10 weeks, it reaches a concentration of 15%, which is amazing because you can imagine at 10 weeks pregnancy, the mother and the baby, the size difference is huge. And yet, the baby is pushing all this DNA out in the mother's blood. Then we thought maybe we can actually use this for some prenatal testing. Now, the number one reason why pregnant women go for prenatal testing is because they're worried the baby might have chromosome disorders like Down syndrome. So I was thinking, can we use this to detect Down syndrome? And of course, normally we diagnose Down syndrome by taking a cell from a baby and then probing it, and then counting how many chromosome 21 we have inside our cells. But a problem comes, in my field, we don't have cells. The DNA just swimming outside cells. So how can you look at some free-floating DNA and try to reconstruct what is inside a cell? So initially, we actually try many different methods, and I'll talk a little bit about those methods later on in the talk, including like how to separate the fetal DNA from the mother DNA, etc. But none of those work very well. So eventually, I thought, is it possible to diagnose a chromosome disorder without separating the mother's and fetal DNA? It's almost like this, like you have a wallet, and sometimes you have 4 coins in your wallet, and some other day, you might have 3 coins. And so I want to know today how many coins you have in your wallet. And of course, one way is to ask you to hand me your wallet, but that would be like invasive prenatal testing. But another way is to ask you to stand on a balance and to see how heavy you are. Now, provided your balance is very precise, the day when you have more money, you should be a little bit heavier. So what we mean is that I need to build a molecular balance or some sort of calculator about how many DNA molecules we have. And of course, up to that point, we do PCR en bloc. We amplify multiple molecules in one go. And then I was thinking, well, how about if I don't amplify everything together, how about I do it digitally? Of course, nowadays, it will be very obvious to you, but it wasn't obvious at all in the middle of 2000. So we're thinking in that situation, if I do many, many PCR, so many that each PCR only have one molecule or no molecule, then in that situation, the fetal DNA would be on its own without interference from mother's DNA. And also, I can arbitrarily increase the precision just by increasing the number of digital PCR. So we did that, and actually for the first time, we're able to show that I can detect a Down syndrome baby from maternal plasma. And so we published this paper in PNAS. But strangely, a few weeks after this paper was published, there's another paper published using basically the same approach. And also very strange is that the 2 papers were submitted on the same day. So of course, what it means, a good thing is that the method probably worked because you have independent reproduction. And the problem, of course, is that there is competition. Now, so this digital PCR method, although it proves the principle, it still have some problem, have some weakness. And one of the weaknesses is that in those days, if you try to do a PCR, of course, I have to find a pair of primers to amplify a gene on chromosome 21, and then another one in a reference chromosome. But the problem is that on chromosome 21, there might be something like 150 genes. And so by targeting just on one, you're basically missing information for the other 149. So I was trying to think, is that possible I could have a method which can optimize or maximize the use of those information in the plasma? Then I thought, well, at that time, the next generation sequencing was just coming in. Then But most people use next-generation sequencing up to that point for qualitative diagnosis, to see if some tumor have mutations, etc. But quantitative sequencing wasn't actually the thing at that time. So I was thinking, is it possible to do it quantitatively and randomly? And for example, we basically randomly sequence millions of DNA molecules in plasma. And I don't have to separate the mothers from fetal DNA. And then align each of the sequence to the genome, and then we can tally the representation of different chromosomes in plasma. And interesting, when we did that, we find it works amazingly well. So here on the left-hand side, you can see in cases with Down syndrome, the signal is very high compared with the normal pregnancies. So we actually tried 28 cases, 14 with Down syndrome and 14 normal, and got 100% accuracy. And so very happily, we published this paper in the subsequent year. And interestingly, within a few weeks of this paper, the other team also published their paper. So then that made me really curious. So I was wondering, who is this guy? And of course, I saw his name is Stephen Quick, who he was already mentioned multiple times in this conference. And then I started to look up his website, and then I realized he's a professor from Stanford. And also, very interestingly, I noticed he actually also went to Oxford, and actually also qualified in the same year that I and my wife graduated, which is 1994. So I went home and told my wife about this. And very insightfully, she actually took out this picture, and that's Steve Quick. So it seems like somehow, fate has draws together. But the story hasn't ended even there, because a few years after this, I was fortunate enough to be elected into the US National Academy of Science. And I remember the day when the result came out, I was looking, and I saw my name. Of course, I was elated. And then a little bit further down, I saw his name. So this was actually taken at the induction ceremony of the NES. So the three of us decided to just replicate that photos. So anyway, so back to the work. Now we have this prototype system, which seems to allow us to non-invasively detect Down syndrome from a mother's plasma. But then to push us out clinically, we need to do a clinical trial. So we spent the next 3 years doing that, and the result was very good. So this was, you can see on the right-hand side, this is Down syndrome cases, and basically we have accuracy over 99%. So I thought, well, it's time to do some pushing out in the clinic. And of course, I couldn't do that in the university. I have to work with companies. And so for the last 20-odd years, we have been very diligently filing patents in this area. But that is now. But in that time, we weren't very experienced. So I remember actually the very first patent we filed, the university actually licensed to a UK company, which didn't quite realize the significance of this work. So they sat on it for a while and then thought it was rubbish and gave it back to us. So, and of course, in a way, it is fair that the manager who made that decision now has left the company. So then I went on one of the conferences, and I met this gentleman who is Charles Cantor, who at that time was the chief scientific officer of Circulating. So we start talking and collaborating, and eventually my technology was licensed to Circulating. Now on the other side, of course, in Stanford, the professor had much more commercial experience than us. So Steve Quick actually licensed his technology to Prenatal, which was then acquired by Illumina. So now basically you have on one side, Circulating got my IP, and the other side, Illumina got his IP. And typically, they use those as weapons and start fighting. So there's a series of something like 4 lawsuits, and eventually, actually, we won 3 of them, and the other side won 1. So we then settle and then pool all the IPs. So the technology is actually now available worldwide. It's called NIPT, non-invasive prenatal testing. Basically, it's become a standard of care, and actually millions of cases was done everywhere. Okay, so apart from prenatal impacting that area, this technology also have impacted in other areas. Now, because you recall that I detect fetal DNA in maternal plasma by using a Y chromosome for male baby. And after we've done that work, I'm thinking, in what other clinical scenario would we have a woman inside her would be tissues from a man? Then after thinking about it for a while, I thought, how about transplantation? And so we actually show in sex mismatch transplantation, for example, with a woman in which a man donated a liver or a kidney to a woman. Then we show that we're able to find the Y chromosome of the man in the plasma of the woman. And later on, we can actually extend that to other bodily fluids, like urine. And now, so we show, for the first time in urine system, that if you have a rejection episode, when there's increased cell death, that actually there's a shooting up of the DNA from a donor, like this. And this has now been commercialized. For example, in the US, a company like CareDx, et cetera, use this type of technology. And then another very usefulness of this technology would be, what is the origin of cell-free DNA? What is the tissue of origin? So we can also use this transplantation method to do that. Now, for example, I want to know what is the plasma DNA coming from. So one way we do that is to do bone marrow transplantation of a man to a woman. In that situation, we find that plasma has basically changed sex from female to male. And then in that juncture, the plasma is male, but the kidney is female, it's from the woman. Then I want to know what happened to urine. And so we did that experiment and showed that urine becomes 70% male. So in other words, some of the plasma DNA can be filtered through the kidney into urine. So by using this sort of approach, we gradually, bit by bit, build out the image of what is the composition of plasma DNA. And then you also recall the reason why I discovered cell-free fetal DNA is because I read about the work in cancer. So now with prenatal testing going strong, I'm thinking maybe we can also feed back what we learned into cancer. And then I want to bring up this slide again, in which initially we try to find ways to separate the fetal from the mother. How do we do it? Then one of the approaches that we did was actually by using DNA methylation. So this work was published in 2002, which at that time was actually, the epigenetics is not as hot as now. And also at that time, we don't really have this good markers which are tissue specific. So how can we do it? So in the end, we decided to use the situation of genomic imprinting. So what happened is this. So it's looking at a particular imprinted region in which a woman have obtained her sequence from her father. And then this region is such that if you obtain from your father, it is methylated. And then the woman then passes it on to the fetus. Because if she's female, then the methylation status change into unmethylated. And then we wonder in that situation, if the baby release the DNA back in the mother's plasma, can I detect that? Can I differentiate this unmethylated A allele from the mother's unmethylated G allele, which we got from her mother? And eventually we do this experiment. So there's a control G allele, control A allele, and the mother's own DNA, and this maternal plasma. So you can see that we can actually see the fetal DNA. So after that, then we thought, well, can I push it beyond the imprinted locus? And then we start to screen a variety of genes. And one of the genes that we're coming across is the maspin gene. Now, for example, here is the bisulfite sequencing result, in which you can see the maspin in the placenta has a lot of unmethylated sequence, whereas in maternal blood, it's virtually all methylated. And then the unmethylation increases with gestational age. And then we show that you can actually detect this unmethylated maspin in maternal plasma, and after delivery, it's gone. And the message actually parallel that of a genomic marker, which is Y chromosome. But the question is, can I then generalize this approach from the fetus into cancer? So then we actually did this. So we did genome-wide bisulfite sequencing of the plasma, and then we actually look at a hypomethylation of the cancer. And also at the same time, we can even look at copy number aberrations, which is essentially the non-invasive prenatal test done in cancer. And you can see in this case, so the outer ring is a methylation. You can see that in the cancer, we have hypomethylation, which is a red signal. And one test, you can do multiple types of cancer, the nasopharyngeal cancer, lung cancer, and breast cancer. And the inner ring is a copy number aberrations, amplification, and deletions. So after that, we can also use this now after that, with the more and more genomic information coming out, for example, from the roadmap project, we also thought about this way of doing plasma DNA tissue mapping. So basically, by comparing the bisulfite sequencing result in plasma with a series of reference methylome, we can work out what's the composition, what tissues release the DNA in plasma. So to localize, where is that tumor? So then we thought, well, this might be a useful technology, so we commercialize this and I found this company called Serena, which is basically to screen cancer early so they can do something about that. But at the same time, Illumina just found a company called Grail, and you can see with exactly the same goal. And so Serena and Grail are going in the same direction. And from my past experience of litigation, I know if we don't do something about that, we'll probably see each other in the court, right? And each time you do that, you probably waste 10 years of my time. And we don't have that many 10 years to waste. So eventually, the two sides decide to talk, and then we merged together. And so I became a scientific co-founder of Grail. And so this was actually taken at the party when we merged together. And then this is Jeff Huber, who is the founding CEO of Grail. And the background is the NBA final in 2017. So basically, after they've taken those technology, then Grail has now launched this test called Gallery, which is based on this epigenetic test and the epigenetic tissue of localization. And recently, actually, there was a study to talk about the performance, which is just published in Lancet Oncology, in which they look at over 6,000 participants, in which the median age is 62 years. And you can see that if the test is positive, the chance of that person having cancer at that moment is 75%. If it is negative, then the chance of that person not having cancer is 97%. And the localization of the tissue accuracy is 85%. But the weakness of the test at the moment is that if you split the data up, you will find that for stage 1, the detection rate is only 24%. They increase to 57% second stage 2, and then 85% stage 3, and 95% in stage 4. So finally, in conclusion, how about convince you, Circulating DNA has created a certain paradigm shift in medical diagnostics. And you can see that there's parallels in between different conditions like pregnancy, cancer, and transplantation. And the NIPT is already a clinical standard of care. And then we have emerging tests in the cancer and transplantation area. And much more work is needed about clinical efficacy of this cancer screening test. For example, how can we push up the detection rate in early cancer? And secondly, by detecting, do you really make a difference? Do you shift the stage of detection? And do you improve the survival? And finally, I'd like to thank individuals for my group for generating the data which I present to you today. Thank you very much for your time.